Hi, my name is Avalon, and I'm a graduate student at Cal Poly San Luis Obispo. My research focuses on HIV, or human immunodeficiency virus. This virus infects millions of humans worldwide and can cause a variety of symptoms that include headache, scratchy throat, spots or sores on the skin, dry cough, nausea, vomiting, and diarrhea, muscles that are sore and achy, thickened nails, or joint pain. This virus works to infect humans by first binding to receptors on the surface of human cells. These receptors then allow fusion. This fusion unloads genetic material and unique special proteins that allow it to change its genetic material from RNA to DNA and integrate into the DNA of the human. It will then use human proteins to replicate and send these new copies of DNA to the edge of the human cell where it can be packaged into new infectious particles of the virus that are budding. My research focuses on this step right here, where the DNA of the virus is floating around in the human cell. In order to avoid being broken down, the DNA must bind human proteins that will help keep it stable. One of these human proteins is called YB1. It must become activated. This is done by a molecule called phosphate. When the protein's in its off conformation, a phosphate can be added to a specific site. Once that bond to the phosphate is made, it will cause the protein to turn on by a change in shape. This new shape will allow the protein to move two nucleic acids, bind, and act in whatever function they're supposed to be. At the molecular level, a phosphate group looks something like this. The reason that the protein changes shape is because of these negative charges that surround the molecule. These charges will cause different parts of the protein to either move closer or move away. In my research, we're working to make a version of YB1 that's always active by adding this molecule where a phosphate would usually bind. This molecule will have similar negative charges and make the protein act as though there is a phosphate in that place. The way that we edit the protein is by the construction of something called a plasmid, which is a small circular piece of DNA. We will take pieces of DNA that code for the protein that we're trying to make, and we'll integrate it into that plasmid to make a whole new sequence of DNA. This plasmid can then be forced into cells by an electric pulse. This pulse will make the cell membrane weak so that the plasmid can move through it. Once inside, the membrane will fix itself. The plasmid that's inside of the cell can be used to express the newly designed protein. The reason for making an active protein is to better understand how this protein could influence HIV. The more knowledge that we have about HIV, the better we can understand the virus and work towards finding a cure.